invitado que tenemos esta tarde obtuvo su grado de doctor en medicina en 1985 de la Universidad Stellenbosch en Sudáfrica. En 1993 obtiene la especialidad en patología en la Universidad de Durban en Sudáfrica, con sus especialidades en patología anatómica y patología química. Durante esa época trabajó en diferentes hospitales en Sudáfrica y en el 1998 incursiona a trabajar en una empresa de venta y mercadeo de equipos de laboratorio llamada The Scientific Group. En el 1999 pasa a trabajar con una empresa francesa que hoy día pertenece a un conglomerado japonés que se llama Oriba, a trabajar con ABX, que también es de hematología, y entre el 99 y el 2003 dividió su tiempo entre Francia y California en los Estados Unidos, que cuando nos llegan a nosotros acá en los Estados Unidos. Y en el 2004 comenzó a trabajar con Sysmex América, ocupando la posición de director de asuntos científicos, o, ¿verdad?, en nuestro español, Director of Scientific Affairs. Así que démosle una calurosa bienvenida a nuestro invitado, el doctor Alexander Guiles.
As you heard, I'm from South Africa originally. I've been in probably 30 countries or more. I've lived and worked in 4 different countries. But I found out yesterday that I'm actually Puerto Rican. And the reason why I'm Puerto Rican is because you can't possibly feel this comfortable and not be Puerto Rican. So I'd like to thank everybody for making me feel so comfortable, and I think you've got a very healthy society, and it's a beautiful day outside, beautiful country, beautiful people, and thanks very much. Okay. So today I'm going to try and explain to you some of the things that we're involved with. When we do validation of instrumentation, we have certain tasks that need to be fulfilled, especially when we're in the United States. We need to be wary of the FDA, and none of us like regulations that much, but the FDA does have a pretty significant role, and I think we can be grateful for those rules being in place. I think they protect a lot of people. We also work with guidelines development. Again, clinicians, myself included, don't like being told how to treat patients, because we should be allowed the autonomy to make treatment decisions on our own, and we always will be able to make those decisions on our own. But there is a need for standardization of clinical approaches, and I think we all recognize the need for standardization too. There's also a lot of changes happening in the environment in which we operate, mainly driven by pharmaceutical innovation. So as a diagnostic company, we need to be very well aligned with advances in pharmacology, and as clinicians we need information relating to the drugs that we use, and we need to be able to monitor both the responses of the drugs, the side effects of the drugs, and also the responses of the disease state. So all of these aspects are important to us as diagnostic companies, but also as diagnostic laboratories, and also as clinicians. And we're all on the same team, because if we deviate from what makes sense for the patient, then we all lose. And if we are very well aligned with what the patient needs, then we all win. So although I represent a diagnostic company, we are all striving for the same end goal. And if we can all look for opportunities to transform and advance what we currently do, then we're doing things right. And so transformational opportunities exist. And we sell primarily cell counters, which, as you know, are screening tools. They're not always definitive diagnostic tools. And so it's the first line of defense that we have, or the first line of investigation that we have, And often we need to go to more specific tests and diagnostic approaches to finalize the diagnosis, but at least with the screening functionality that we have, we're the first ones that get looked at. And because Sysmex is #1 worldwide in terms of the number of cell counts that are done, Sysmex is probably #1 in terms of the number of samples that are thrown onto our instruments. So cell counting is not particularly interesting in terms of picomolar quantities of protein, but cell counting is interesting because of the scale of the testing that we do. And the most common tests that we do in clinical medicine are related to cell count. So anything that we can do on a cell counter to enhance the analytical and diagnostic performance will have an impact. And there's a lot of advancement that can still be made, and many of us don't recognize that because we're satisfied with the way things are. And in diagnostic medicine and in medicine in general and in everything that the world does, change is constant, and unless we're advancing, we're not doing what we should be doing to move forward. And so one of the other things that we're doing is working in the preclinical arena. We've got some instruments that have veterinary capability, and when people are doing clinical trials and they're looking at animal models, we have instrumentation that can do similar things to the changes that we're making on the clinical side. Okay. Sorry. So the efficiencies of the systems that we make are primarily the reagent that we use. We use reagents that have non-specific staining of nucleic acids. So nucleic acid-rich components of the cells will be fluorescent. And it's not that straightforward, but on top of that, we also have the differential lysing of the membranes. And so different cell types have different reactions to the lysate. And some of the cells have big holes in them from the lysate, and some of them have less big holes. And so the staining rates are differential for the different